afternoon and welcome back at the LabAutomation online days. We've had a busy day so far with lots of interesting webinars. And we will continue now with Nathaniel Hentz from Artel. You can see him right next to me in the screen. And he'll be telling you all about Pipette tip performance verification. Marcus from Analis is also available in the chat to answer any questions you might have. So please don't hesitate to ask them in the chat screen. Then Nathaniel, go ahead.
All right, thank you very much. Welcome and good afternoon, everyone. It's morning for me, as I'm from the States right now. But as Elena mentioned, I'll be talking to you today about disposable pipette tip verification. And, you know, I have this term guided by science. I think we all guide our things by science, but we'll, I'll illustrate a little bit more detail what I mean by that. So today we're going to be talking about optimizing a workflow process, so, quick introduction on that, and then we're going to talk a little bit about sourcing disposable pipette tips. As we all know, it's not unique to one company or one manufacturer. There is a shortage of tips out there in the world as the field starts to ramp up the manufacture of these. It'll be getting better, but we'll talk a little bit about some of the constraints there, and then probably spend a little bit of time on explaining why the pipette tips are so variable. We'll talk a little bit about the manufacturing process and then about the fluidic properties, and then I'd like to spend the rest of the time talking about using Artel MVS to evaluate these pipette tips. And this is in the absence of biology, so you can set up a nice pipette tip verification program. I'll explain the methodology, and then we'll take a look at some example data analysis. And again, these are some kind of suggestions that we'll talk about, and then we'll wrap it up with a summary and open it up for discussion. And again, as Elena mentioned, I'll be here for questions, and then Marco will also be available for questions. Just real quick, in terms of an average assay workflow, Artel, we've had this tool, the MVS, the multi-channel verification system that we've had for a number of years. It was dedicated primarily to instrument calibrations. But along the way, we have developed a number of applications that take advantage of the unique situation or the unique technology that makes up the Artel MVS. And when you think about a workflow and an automated assay workflow, it's really a process. And if you take a look at the diagram to the left, we have a number of applications that address various parts of this assay workflow. We've got liquid class optimization for the liquid handler, labware qualification, pipette technique training, mixing, washing, and then again the instrument calibration. And why I bring this up, it's very important when we think about automation, when we're especially considering transferring a method or even developing a new method. So this would be going from manual to an automated platform, or from automated platform to another automated platform in a different laboratory. So for today, what I'm going to do, instead of talking about all of this stuff, we're going to focus on the labware qualification portion of it, and very specifically I'd like to address the disposable pipette tips, and then again we'll talk about that in a little bit more detail. This is not news to anybody. There is a pipette tip shortage out there. We all know this. It's been well reported. We all feel it in the laboratories. And just a couple things that I do want to bring up here. We know that manufacturers are ramping up capacity. Labs are right now going to alternate sources, or they're stocking up, or maybe have stocked up. A lot of labs are reusing tips for their automated platforms. And we also know that quality is critical. And this last bullet point here, I can't overemphasize how important this is. I think a lot of times people think that the pipette tips, no matter where the source, if they're cheap, that's better, and that they have the same quality. And really, what we're going to talk about here is that they don't, and you need to pay attention to that. So again, when people are either new to automation or are in the automation realm, and you're going through many tip types, and you start thinking about going to alternate sources, we need to really pay attention to the quality. Right now, again, customers have plenty of options. People are switching tip manufacturers. And again, when we do that, it's really the differences in manufacturing quality that varies a lot. There's a lot of good manufacturers out there, and there's a lot of maybe not so great manufacturers. And it's really differences in the raw materials, and we're going to talk about this. And all of that is I want to really illustrate here. It has an impact on your volume accuracy and precision. A lot of people are switching over and doing the clean and reuse method. So these are the existing tips, and this has been employed for years in the automation realm. It's especially when people will take the tips and just do a simple water or detergent wash, but people are paying more attention to this now because of the shortage of tips. And again, it's you have to think about the effect of the cleaning on the volume delivery, and then when you reuse these tips, you have to remember that these tips are constantly being reseeded and removed from the mandrels of the automation system. So that's going to have an impact. And then if there's any kind of a buildup of cleaning agent, that's if we start thinking about detergents and things. So it's not just about cleaning and then reusing and everything is fine. We see a lot of people that are buying larger inventory when that's possible. You either buy multiple lots of tips. The thing that I want to really talk about here is the tip storage and tip quality. And when people in the beginning of this situation, or even if it's opening up, people are still buying large lots. And if the storage of the tips isn't proper, if it's depending on the conditions, you can alter the quality of the tips, and that's really around the structural integrity. So just a few things to keep in mind. Where does this matter? Pipette tip verification, it's pretty much everywhere. I have, a lot of bullet points here, but it's where people are using a large number of tips. And, again, we see this in the clinical diagnostic labs, especially those that are running automation, clinical genetics testing labs. And we've seen an explosion of these types of labs with COVID. All the CROs, CDMOs, manufacturing QC labs. So anywhere there's automation or where quality is important, this is where it really makes a difference. So let's talk a little bit about the liquid transfer. So we have our pipette tips, and there's many things that affect the liquid transfer, and I'm going to talk about a couple of things, not all of these, but let's talk a little bit about the liquid properties so we have a better understanding. So when we think about the liquids, there's density, obviously, and that's just it's a weight per volume. And again, that's going to have an impact on especially delivering automated delivery for liquids for your assays or samples if they have to do some kind of sample prep. And I bring these up because when we do manual transfers, we have we automatically adjust on the fly. We have this innate sense. We're looking at the solution. We can see that we need to pipette slower or faster. But when we think about an automated system, we've got to program it to do that. And so when we have differences in density, if we have differences in vapor pressure, and all of these things can have viscosity. An automated system can't automatically adjust. So we have to be able to, anticipate the solutions that we're dealing with and really understand their behavior. Surface tension is another thing. And again, these are detergent type of solutions. The viscosities, maybe the glycerol PCR mix, if we think about that. So there's a number of factors that, regardless of the tools that you're using, that these are going to affect the dispense. And so a couple All of this with the components, and I want to spend a little bit of time here because this is what a lot of times people don't really lend a lot of thought to, and they just use the plastic tips. They're not created equal. We all know tip size and volume. Obviously, that's going to be a different performance. The tip type and the material, manufacturer cost, availability, and all of that. All of these things make a difference, and we know that. But what I do want to spend some time on here is let's talk a little bit about the manufacturer on the next couple of slides. And the other thing to keep in mind is just a little piece of advice here. When you're thinking about using or employing tips, even if it's new, if you just started a brand new assay and you're deciding on what tips, I would highly recommend performing a tip study when picking whatever type that you're interested in. And then the other thing to keep in mind is the automated liquid handling manufacturers, they typically only provide support when using their tips. So if you have a very good reputable manufacturer of a universal tip type, a lot of manufacturers won't support the application. So keep that in mind as we're moving forward. So now let me spend a little bit of time on the components, and we'll talk about tips very specifically. We've got the standard tips. These are designed for everyday lab work. That's what we use in the handhelds, and most automated systems use those. And then we've got the barrier or the filter types, and those are if we're trying to minimize contamination. Regarding samples, or we've got viscous materials, those types of things. So those are the two more common tip types. Another common one is the sterilization of these tips. So these are pre-sterilized. They're designed for cell work typically, and commercial tips, they're designed for if they're pre-sterilized, they're designed for this. And I do want to bring up one thing here. I see a lot of labs when they, especially during the shortage, or even not, they'll buy tips and then they'll do a sterilization in house. They'll do typically an autoclave cycle, and while that will sterilize the tips, we have to remember that most tips are made of polypropylene. And they become soft at about 110 to 120 Celsius, and they will melt at 160 to 180. And so we have to think about the standard autoclaving conditions. We're basically softening up these tips. So we have to keep in mind, if we're going to autoclave according to standard methods, it's going to stress the material. So what that's going to do is potentially alter the fit on the automated system, the mandrels, the 96, 384, or 8-channel, depending on the device. So keep that in mind. It really is better to buy pre-sterilized because that's either gas or gamma radiation. Some tips are low retention by design. They're treated to become hydrophobic, so that's going to have an impact. So those aren't interchangeable with the above there, the non-barrier standard tips. They do have a difference in behavior. So keep that in mind. Graduated tips, they have little markings, and what that can do is afford places for liquids to cling. And when liquids cling, you form little droplets, and then sometimes you can over dispense unbeknownst to you. And then there's these ergonomic tips. We don't typically have to worry about this for automation systems, but manual, I do want to bring this up. They're designed for lower ejection forces. So when you click them, they pop off very easily, and it's really to reduce the repeated stress injury. So, but again, they're not very good for repeated use. But again, this is more for manual, not necessarily automation systems. And why I bring all of that up, we've got to think about the fit. And the question that you need to ask yourself is when you're buying a universal tip is this truly universal? And really there are no truly universal. It's about the fit. And it's if you have to jam the tips on, if you have to set a higher force on your automated liquid handler to get some universal tip to seat well, it's probably not the appropriate tip. And a lot of times you can take these pipette tips and lay them on the counter and see if they wobble. If they do, that's probably not the appropriate tips. Sometimes sealing rings can be used to help with that, to form that seal, and that can be corrupt too if it's not a perfect fit. So just there, there are a number of factors feeding into the performance here. And as I promised, we're going to talk a little bit about the pipette tip materials. Stainless steel, either native or Teflon coated, those are available. A lot of times those are used to pierce septa, to pull samples out and then reformat them. But by far the more common ones are these polypropylene. And these are the ones that really vary by manufacturer. Some people use mold slip agents, biocides, residual plasticizers from the manufacturing. Sometimes there's hydrophobic additives like silicone. And the issue here is this is if you go to reputable manufacturers, these are less of an issue. But if you start going to the real cheap pipette tip manufacturers, there's a lot of companies that are springing up right now to address the shortage. We have to be very careful of whether it's a virgin or recycled polypropylene or whether some of these agents are being used, because they have a direct impact on the performance. Even if it may fit properly, it's the liquid inside that's being delivered. The other part of it that we need to talk about is there is potential to affect the biology, the assay biology. We're here to talk about pipetting performance, but it actually can affect the assay biology. So be aware and keep in mind that there are a number of factors at play here. I've included this quote here, just so we can at least talk about it. It's not just me that's saying this. This is pulled off of the Mettler Toledo website. They talk about pipette tip purity, but it really does affect the accuracy of the data. And what you don't want to do is waste time when you can do something about it right up front. So again, there is a potential, and this happens actually quite frequently, wasting weeks, sometimes months. You're going down a series, a whole campaign of screening or testing, and then you realize that there's a drift in the performance of your assay, and a lot of times it can be traced right back to this. So let's address this right up front, and that's really what this webinar is about. I do want to point out here that the good reputable dealers and manufacturers of pipette tips, Sartorius is an example here. I'm not a proponent of using them necessarily, but this is a good example of their quality certification, and that's really what I wanted to show here. Is they'll talk about all of these. They'll say that they don't use mold slip agents and what type of polypropylene is used, and they'll describe all of that. And sometimes they'll do purity testing if you're working on manufacturing QC and you don't want endotoxins or RNases and DNases. So, you can get these performance or these quality certifications, but there aren't necessarily anything that deals with the performance of the tip. So this talks about the components. What we're going to talk about in the next couple slides here is really about the performance of the tips. And what I'd like to show here is we can very clearly see good tip and bad tip. And really this is about all of this flashing material. If it's non-centered, if it's rough edges, these are all places that can attract liquid that you don't want. So it can either bring in extra liquid, extra volume just through the capillary action, or sometimes it can retain fluid and give an under dispense. So again, this is about fit and it's about liquid delivery. So let's talk about designing an appropriate tip performance verification program. There's really three components here. It's the measurement, And that's really composed of the assay. And then I've got in here as a second bullet point, the Artel MVS technology. And we're going to talk about the advantages of that in just a second. And we want to design the appropriate work plan. So that's the frequency, the number of replicates, and then the analysis tools, how it is you want to analyze your data. And then this is the part that a lot of times people forget. It's designing the specifications. Again, this is based on data. Both MVS and your assay. So we'll talk about that in the next couple of slides here. I mentioned the Artel technology. It's seemingly simple, and you know, technologically it is. It's a dual dye, dual wavelengths absorbance measurement system. And if we take a look at the graphic on the right-hand side, we see the red dye being dispensed, and that's really the quantitative part. And then we've got the diluent, and that's the blue material. That's the non-quantitative part. And what that does is it brings the volume up all to an approximate equal level. So what you're doing here is really quantitatively measuring the red, and the blue dye is really giving you that path length determination. And then you measure the two absorbance units, or the absorbances at both at the red and the blue dye. And then through a derivation of the Beer-Lambert law, we can actually calculate the volume of the sample. And what's nice about this is a biological independent method. This is looking at strict performance of the tips. So we use it primarily. A lot of customers use it primarily to determine the volume dispensed, but you know, the pipette tips are actually a vital component to that. So we can actually use this to qualify tips, and we'll talk about that in the next couple of slides here. This is the basic workflow. You dispense your red solution. If you look in the upper left-hand corner, and then you move over to the right, you add your blue diluent. There's a mix on the validated shaker, and then there's a plate read. Again, we look at the two wavelengths, and then there's a report set up. And then what's nice about this is it'll provide dispense accuracy and precision. It'll give you statistics for the entire plate. You can do channel by channel, and then dispense order. So depending on the information that you're interested in, you can extract it out. So it's very easy. It's a couple-minute read. And the important part of it is if we take that and extrapolate into looking at tips only. So I'm showing this quick example. If we look at the plot to the left, what we're doing is comparing two current lots, 1 and 2. We're looking at the performance, and now we're evaluating pipette tips from a new source. And very, very clearly we can see lots 1 and 2 are behaving very similarly. But from the new source, it doesn't meet our specifications. The variability is way too large, and the accuracy is not within the range that we want it to be. So what we're saying here is, just because the new tips, they're 30% less cost, which is fantastic, the performance of these are not meeting our expectations. So again, if we think about these are just dyes that we're working with here. If the performance is not working out with the dyes, can you imagine what it's going to be like when you start compounding the biological variability here? And this is typically what happens when you have a method transfer failure. A lot of it can be traced back to the sources of the tips. So again, keep in mind we have a standardized method for qualifying tips before they get into your assay workflow. So let's spend a little bit of time thinking about setting up a program. As you're looking at identifying tips for your automated assay, you want to consider the volume that's going to be used and versus the tip volume. So if you have one volume that you want to dispense, that's pretty easy. You select a tip where it's going to be maximum of takes up most of that volume. But if you have something that you're going to be doing a whole range of volumes, you'll need to select the appropriate tip, and then you'll need to qualify those tips for what you're interested in. And again, you want to make sure that you're identifying how these tips are going to be used in your automated assay. Are you going to be using them once and discarding, repeated use? So when you're setting up your verification program, you want to replicate that. Do exactly what you're planning on doing in your automated assay, and then you can set up this verification program. And again, I have that last bullet point here. Make sure you specify your pass-fail criteria. A lot of times people forget this, and they just look at statistics and they don't really have anything to compare to, and so that makes it difficult. So just make sure you understand. Design your pipette tip verification program appropriately. This is an example here. If we have this is a fictitious liquid handler, the Dispensomatic ALH96. If I were to look at my current tips versus new tips, this is how I would set it up. I would set up 5 replicates, 5 plates worth of the low volume. That's 10% nominal volume, and then do that. Compare current tips and new tips, and then if you look at the right-hand side, you do the same thing at the maximal volume or the nominal volume. And then do the 5 replicates, and then you would go through your data analysis. So you would use these in the MVS workflow, but use it, everything's the same except for the tip types. And again, in this example, you'd be generating 20 plates worth of data, and then do your analysis. Speaking of analysis, what is it you would look for? And I do want to talk about this term statistically different versus experimentally relevant. We could do the typical volume, accuracy, and precision. That's great. We can do column, row, plate, the overall replicates. We can look for, and I would highly recommend, trending and patterns, as would be plate to plate, tip to tip, lot to lot, and numbers of reuses. So if you're going to use tips 1, 2, and 3 times after cleaning cycles, let's say, you'd want to make sure you replicate that and look for any kind of trending. And then analysis methods. You can use your traditional statistics, mean, standard deviation, percent CV. Statistical process control charts are very useful in this type of analysis. You can use analysis of variance, Welch's t-test, or there's other statistical models that you can use to compare lots, tip manufacturers, or effective washing and reuse. So I'm not going to tell you which one to use. That's what you have to determine, but I'll give you a couple of examples on how we would go about this. And this is this idea of statistically different versus experimentally relevant here, and I'm showing this graph here to the left, with the intent of not even showing you the y-axis, and we can see that they appear to be 2 different populations of tips. The new tips are behaving different than the current tips, and what I do want to show here is if I put the scale on here. What we actually see, and we do a statistical analysis, the means are different. According to this particular analysis of variance, but the variance isn't different. So what we have in this scenario is the mean for the new tips is only 0.1 microliter difference. So this is where statistically it might be different, but experimentally, is this going to cause enough of a variance in your assay data? And that's what we also have to consider. So it's not enough just to do a statistical analysis. You want to run this first and then run your assay and compare your uncertainty values there. So again, you'd want to run this because this is a very quick test, and then put it in the context of your assay. Trending, scatter plots are very useful for monitoring trending. As we can see, I've got current tips in the orange, and then I've got the new tips in the blue dashed line. And what we see here is they behave pretty similarly. We've got a downward trend going across each row. But again, let's look at the left axis here. Is that meaningful in terms of an assay? We see the rightmost column of tips I've got circled here. Those happen to be, they appear to be the lower dispensed volumes. But again, we have to keep this in the context of the assay. Is this going to affect our overall performance? So again, we can look at trending, and these are really nice to keep an eye on, but we also, if I keep repeating myself here, I'm doing that on purpose. Keep it in the context of your biological assay. And this is where it's very useful if we use statistical process control charts. Like in this case here, what we see is tip batches 1 through 5 are behaving almost identically, and then 6 through 10, or maybe 6 through 11, we start to see a decrease in the overall volume. So while it really hasn't caused a problem yet, it's still within our upper and lower control limits. They're definitely, it's something to keep an eye on. So we would probably have what we would call an alert limit. And then we see here on batch number 12, that exceeds our upper control limit. And we'd want to stop. We want to intervene, get rid of those tips, or evaluate and see if there's, and do some troubleshooting on that. So again, this is a very handy tool for monitoring, over time, batch to batch, batch to batch. The other interesting thing here, and this is real data, by the way. So we have an automated liquid handler that's not behaving properly. As you can see in the current tips, there's some outliers, but the new tips, we see that it's a much higher variability. So this actually happens to be more sensitive to the variability in the liquid handler. So sometimes, if you were to qualify these tips and not realize it, you actually could go on and really follow up your assay. So again, suboptimal automated liquid handler, it may be more sensitive to the different tip sources. So make sure that you qualify them. Don't just do this on a liquid handler that works perfectly. You want to do it on the liquid handler that you're going to be using in your automated experiment. And I see we're almost at I guess we're at our 30-minute time, but we're about ready to wrap it up here. I just follow up with a couple of key elements here to think about. On the manufacturing side, if you're a tip manufacturer, this is a good way using Artel MVS to develop a robust QC program. And you can generate data that end up creating a certificate of analysis, much like you would for purity and such. And again, you can get lot-based performance certification. And for people that are new to the manufacturing, it's a way to differentiate yourself from other manufacturers. And then for users, that's us. That's the folks that are in the lab. And again, this is about making sure we're using the right tips for our needs. And we can, evaluate different manufacturers, but we can use science, not just cost. We can use science to compare these different sources. So again, it's a good way to look at alternative manufacturers, but it's also a good way to prevent catastrophes in a screening campaign or a sampling campaign by using multiple lots throughout that experiment. And then finally, the last slide here. Again, MVS technology. It's a real easy, fast way of qualifying tips. It's a matter of minutes. And if we compare this to an experiment, we could run an experiment. But if you think about the average experiment, a qPCR test, that'll take 40 minutes to an hour. And an ELISA will take 4 to 6 hours, depending on the thing, or a next-gen sequencing that could take a couple of days. So, before you get your results. So really, this is a quick way of identifying whether the tips are going to be working in that particular scenario. And again, setting up a Pipette number 2 here, a verification program, it can be fairly straightforward. We don't have to overthink it, but if you use the right tools, like the Artel MVS, and again, standardization, that's the key. So you can take that and go, implement that lab to lab and get the same results over and over, comparable results. And we've talked about this, the quality of the tips, they will affect your assay. That is, that's a known fact. There's just a number of factors to consider, not just cost or availability. We need to make sure that the performance is appropriate. And we're under our 35 minutes here. I would like to thank you for spending your afternoon, at least these last 30 minutes, talking about this or listening to me talk about these challenges and ways to overcome them. And I would like to introduce my colleagues at Analis. They would be your local contacts to talk about Artel MVS and employing that in your automated workflow. Marco and Hans, they're really nice. They're great colleagues of ours. And if you have any technical questions, you can certainly reach out to me, and then we'll get in touch with Marco and Hans. So with that, I'd like to conclude. And again, thank you for your attention.
Well, thank you very much, Nathaniel, for your webinar. I just checked in the chat, and I don't think there are any questions at the moment. So in case there are questions, please drop them in the chat. And in the meantime, I would just like to point out that we are continuing with the last webinar of the day at 4 p.m. That's a webinar by Nasa Bouafit from PHC Europe. If you haven't signed up yet for the webinar, you can still do so via the link popping up in your screen. We'll start that right now. And then this webinar and all other webinars today have been recorded and will be available to watch as a replay. And you will be informed via email when those are available to watch. Quick look at the chat one more time. So if you do have questions, of course, like Marco said, you can always contact Marco directly via email. And then thank you again, Nathaniel and Marco. Thanks again for all the viewers to watch. And I wish you a great day.
Thank you.